Okay, first of all, we look at this, the rate of photosynthesis. We do know that it determines by the three main external factors here: light intensity, carbon dioxide concentration, and temperature. And again, I said that don't forget we have learned about the absorption spectrum and also the action spectrum. So it means that we can fix the light intensity, we fix the carbon dioxide concentration, we fix the temperature. What we change here, we actually manipulate the light wavelength. For example, we can start at 400, 410, 420, 430, and so on. Are you clear? So at the end, you have a range of the wavelength, and we look at which wavelength actually gives the best rate of the photosynthesis. Okay, we do means I've learned that parts already. So in this topic, we won't cover the wavelength, but wavelength we cover the absorption spectrum and action spectrum. Okay. So at any one time, the changes in the limiting factor, when talking about limiting factor, is very, very crucial. The understanding here is when we change, okay, for example, let's say light intensity, carbon dioxide concentration, and temperature. If you suspect that, oh, light intensity is a limiting factor, what we're going to do is we increase the light intensity. Are you clear? Okay, so if we increase the light intensity and you show that, yeah, there's an increase in the rate of reactions, then we confirm light intensity is a limiting factor. This is how a general way we find out whether which factor is a limiting factor. Are you clear? Okay, so it's similar to the wooden buckets. Okay, that is a pail or buckets. So something like this. Nice. You try to imagine. This is a wooden bucket. Yeah, but so it's like something like a wooden plank. That's the one that formed the bucket, say something like that. Okay. So you try to imagine that if this represents the light intensity. So how much water can be held by this container? It depends on the height of this wooden plank. Are you clear? This wood, the height, right? Okay. So if let's say this is the light intensity, and this is CO2 concentration, this is temperature. So no point you increase the CO2 concentration, increase temperature, but light intensity remain the same. Cannot. The volume of the water now depend on who? Depend in the one that actually in the shortest supply. Are you clear? Okay. So in this case, how you increase the rate of reactions? We're going to increase the height. Let's see that. But any further increase later, you can see that there's no change in terms of the volume anymore because other factor now act as limiting factor. It can be CO2 concentration. It can be the temperature. Are you clear? So that is the concept of limiting factor. So it means that if you suspect that light intensity is a limiting factor, you change it. You may increase. When you increase the light intensity, you see that whether the rate increase or not. If the rate does not increase or didn't show any increase in terms of the rate, then we know that it cannot be light intensity. Then we try with the CO2 concentration. Are you clear? Then we try with the temperature until you find out which one is the limiting factor. Okay. So as photosynthesis is a complex process, so means that the entire process, okay, whichever step that is proceeding at the lowest rate at a particular time. Okay, so it's something like the whole class. Okay, the whole class in this case, okay, it doesn't depend on how many of you we talk about pass rate. It doesn't depend on how many A in this case. If the whole class everyone get A, only one student fail, the pass rate will be, it won't be 100%. So it depends on the lowest one, the lowest supply. Are you clear? Are you, yeah, in this case, so the rate of photosynthesis always depends on the lowest supply. If the light intensity is not enough, then light intensity is the limiting factor. Are you clear? It always depends. So it don't care, it doesn't care who is the highest supply. We don't care. Highest mark, we don't care because we talk about the pass rate. So the whole class pass rate, it depends on everybody. So those the lowest value or the lowest mark in this class will determine whether the whole class pass or you can't get 100% pass rate. Are you clear? Okay, so that's the idea. So in this case, that step, the particular step is known as rate limiting step. Are you clear? Rate limiting steps. So let us look at this diagram. So how we interpret this diagram? First, if I start with because my independent variable is the light intensity. Always remember, if I have my independent variable is light intensity, so it means that the other factor I have to constant them, cannot. They are controlled variable. You cannot change light intensity, you change the CO2 concentration, you change the temperature every time. How you know which one actually is the one that gives you the effect? So once you change one parameter, the remaining parameter you have to constant them. So same goes with this experiment. What we change here is the light intensity. What's remain constant will be the CO2 concentrations and also the temperature. Let's say I started with low concentration of CO2. Let's say I get 0.03% CO2. Temperature I set at 20 degrees Celsius, for example. Let's say, okay, 0.03% of CO2 concentration and low temperature. And we do know that now my light intensity, when I increase the light intensity, the rate of the photosynthesis increase, right now. But after that, plateau, level off. Level off why? Means that now I change the light intensity. I keep on increasing the light intensity, but there's no increase in terms of the rate. So it means that light intensity no longer acts as a limiting factor. It may be CO2 concentration. It may be too low the temperature. So which one? You want to find out, we change another factor. Are you clear? So now from the first graph to second graph, what we change? We change the concentrations of CO2. How we change? We increase. For example, 0.03%, we actually change it to higher, let's say 0.3%. But can I change the temperature this time? Cannot, because I change one factor, I have to make sure another factor remain constant. So that's why temperature now remain at 20 degrees Celsius. Can you see that the rate initially, maximum rate is here, right? Now maximum rate higher. Are you clear? But again, in this case, can I get higher? Can. Now you can actually change the temperature. Perhaps you can change from 20 degrees Celsius to 25 degrees Celsius. And then you can see that the graph going up again to a higher rate, but again plateau. So I have changed the concentration of CO2. I've changed the concentration of, the temperature already. So what else are the factors that make this level up? So we talked about external factor, 3 of them already, right? Light intensity, CO2 concentrations, and also the temperature. So means that the internal factor is the one now becoming the limiting factor. No point I give the high light intensity, no point I give very high CO2 concentration, optimum temperature, but the number of the chloroplast is so small. Can I see that? So means that what contributes, what are the factor that contribute to the level of here, constant here, it might due to the internal factor. For example, the number of chloroplasts, the number of palisade mesophyll cells, the number of pigments. Can I see that? Or it might be the wrong wavelength. Are you clear? By right, we know that plants, okay, green plant, particularly with the abundance of the chlorophyll, okay, chlorophyll molecule, then they absorb that. Violet, blue regions, 400 to 500. Red regions, 600 to 700. So, but I shine the light, that's actually 550 nanometer wavelength. That's why the rate constant. If I change the wavelength to the suitable wavelength, I might get even higher rates of photosynthesis. You get the idea? Okay, so how to interpret this graph? So first, because increase. Okay, because we change from 0.03% to 0.3%, it showed that increase in terms of the rate. Can you see that? The rate increase. So that I can conclude CO2 concentration actually is a limiting factor. But if I change, try to imagine, I change from 0.03% to 0.3%, but the rate still remain the same, no change. Then I can conclude that CO2 is not a limiting factor. Temperature. Are you clear? Okay, so this is how we try and arrow to find out who is a limiting factor. Okay, so we start the first one. So we explain now. We have to explain the effect of the light intensity on the rates of the photosynthesis. You can look at this graph. This graph actually reports the rate of photosynthesis versus light intensity in a relative unit. You can write down for the light intensity, a few ways we can change the light intensity, because in paper 5, you need to describe the experiment. You cannot say that change light intensity full stop. How? So few ways. The easiest way, we always change the distance of the light from the plant. Okay, so the light intensity is inversely proportional to what? Inversely proportional to d squared, distance squared. Are you clear? Okay, can write it down. So how we actually measure the light intensity? We can actually do this. So the light intensity, okay, inversely proportional with d squared. Okay, the distance squared. Okay, or we can change the power by adjusting the resistance. The higher the resistance, the light intensity will be lower. Are you clear? Okay, we can change the resistance of or change the current. Now, in this diagram, you can see that first increase and then level off. So how we explain why increase? Okay, so one of the main problems here in the question, most of the time they didn't tell you actually what they measure. Rate of photosynthesis here. Rate of photosynthesis can actually measure by 3 ways. Right now, 3 ways. What are the 3 ways we can measure rate of photosynthesis? First way, we measure how fast the O2 is released. Okay. Second way, we measure how fast CO2 consumption. How fast the CO2 consumption. What's the third way? We measure, okay, the mass, how fast product can be formed. Let's say mass of product. How much sugar is formed. Are you clear? So 3 ways, guys, 3 ways. So generally, if the question didn't say anything, you can actually define your own way in terms of the rate of photosynthesis. But generally, we will use the number of product, because photosynthesis, CO2 and O2, O2 is a byproduct of photosynthesis. CO2 the plant consumes. So the best way to determine how much photosynthesis takes place, then we actually refer to the how much product is formed, and it affects by the, or it determines by the Calorie cycle. How much product can be formed determines by how fast Calorie cycle can take place. So in these explanations, I will talk about the Calorie cycle more. Are you clear? Clear? Now, if you want to go into specific in terms of the light intensity, which pathway light intensity have the biggest or major effect? Light dependent or light independent? Light dependent. Okay, always remember for light intensity, light dependent. Which pathway? Cyclic or non-cyclic? Non-cyclic. Okay. So the major effects of the light intensity actually act on light dependent reaction, which is non-cyclic photophosphorylation, which is part of the light dependent reaction. Why non-cyclic? Because when talk about the Calorie cycle, when you reduce the GP to TP, during this process require reduce NADP and ATP, and these 2 items are produced during non-cyclic photophosphorylation. Are you clear? So can you link now? So when talk about light intensity, so which pathway is affected in this case? Non-cyclic photophosphorylation of the light-dependent process, okay, or reaction. So how it explains? Very simple. So at low light intensity, the rate of photosynthesis definitely is low, but when the light intensity increases, when you increase the light intensity, you can see that the rate also increases. So this indicates that the light intensity acts as a limiting factor. Are you clear? So somewhere, if you look at the graph, guys, somewhere here, about 6, okay? Less than 6, light intensity actually acts as the limiting factor. Okay? So how you explain? So this because at low light intensity, there is a shortage of the products of light-dependent reactions. Okay, due to what? When the light density is low, the absorptions of the light also will be low. Cannot. Can you see that? So photoexcitation rate also will be low. Okay. So which are in this case, because of the light dependent reactions, or put a brackets, more specifically in this case, product of non-cyclic photophosphorylation, therefore reduce NADP and ATP in shortage. Are you clear? Now, if these two items in shortage, which reaction actually will be affected? Which reactions? So the rate limiting step now is the reduction of the GP, where reduced NADP and ATP are needed. So rate limiting step here is the reductions of the GP to TP. Get the idea? Okay. So now, what do you expect to see the concentration of GP and TP in this condition? When the light density is low, what you expect to see the concentration on GP and TP? Yeah, GP will be high, TP will be low. RUBP? Low or high? This is a common question asked. When the light intensity is low, we do know that the step limiting, okay? Rate limiting step is the reductions of the GP to TP. So GP confirm high concentration, TP confirm low. So that's why product is low. How about RUBP? High or low? So it depends. As long as CO2 concentration is not a limiting factor, RUBP continue to take the CO2 until RUBP no more. Because TP is low, after regeneration, everything, so it's something like jam where? Jam at the GP. So GP is high because why? RUBP react with CO2 to get the split and get two molecules of the GP. So GP concentration will be high, RUBP will be low, TP will be low. Why I see some of you don't know what I'm talking about. Calorie cycle, guys. That's frighten me. Okay, RUBP in general. Take in CO2, you get the two molecule of GP. GP now get TP. Cannot the reactions. Okay, so it means that the rate limiting in this case here, this process, because we don't have the reduced NADP, we don't have ATP sufficient. So therefore, this process can take place. If this process can't take place, so confirm GP concentration will increase. TP concentration will low. And RUBP, because in this case, as long as CO2 is there, RUBP will continue to react with CO2 to get GP. So that's why GP concentration is high, but no regeneration RUBP. Can you see that? Are you clear? Okay, so please think about this. So as light density increases further, the rate of photosynthesis becomes maximum and constant now. So we do know that light density no longer acts as limiting factor. So other factors, for example, maybe CO2 concentration too low, temperature not suitable. Therefore, they are the limiting factor now. Are you clear? Okay. Now the effect of CO2 similar. Okay, the effect of CO2 similar. As the light intensity. Okay. Also plateau. So what is the explanations here? So at low CO2 concentration, the rate of photosynthesis increases when we increase the carbon dioxide concentration. But provided that light density is there and temperature, they are not the limiting factor. Okay. So this indicates that the limiting factor is the CO2 concentration, because when you increase the CO2 concentration, you can see the rate increase. So definitely CO2 is a limiting factor. Okay. So what is the rate limiting step? The rate limiting step here, because CO2 involved in the Kelvin cycle. Write down the direct effects from the CO2 is Kelvin cycle. And which step of Kelvin cycle? CO2 fixation. Are you clear? CO2 fixation. So as a result, NADP or reduced NADP now accumulates. Why? Why reduced NADP accumulates? So in this process, if CO2 concentration is a limiting factor, what is the concentration of RUBP? High or low? RUBP concentration will be high. GP below, TP low. Are you clear? Because the rate limiting step here is CO2 fixation. So logical thinking, CO2 fixation stop. When the CO2 fixation stop, RUBP confirmed will be high. Stay there, do nothing, cannot do anything. So therefore, GP concentration is low, TP concentration will be low. Are you clear? Okay, but RUBP, because TP, if any TP there, TP will be regenerate back to become RUBP. So RUBP just accumulate there. And reduced NADP also will be there. No one actually regenerate the NADP. Are you clear? But if we increase the concentration of CO2 until a certain moment, we see that the maximum already and become constant. And we know that carbon dioxide concentration is high, they are no longer act as limiting factor. Maybe temperature, maybe light density is not suitable, or maybe other factor, for example, the wavelength of the light. Okay. Now, when we talk about the temperature, temperature, then you can see that the graph actually very similar to the graph that we have learned in enzyme. So in this case, what is the rate limiting factor? Enzyme denaturation. Are you clear? It depends on the enzyme denaturation. Okay. So one thing that we need to understand here is when we talk about temperature. In this graph is roughly about 30 or 35. Okay, 30 or 35. But in fact, most of the plants will be roughly about 25 to 30 degrees Celsius. Are you clear? Okay, the optimum temperature for the plant enzyme generally 25 to 30 degrees Celsius. Okay, and which pathway affected by temperature? Now, this part students always confused because we do know that, okay, for the photosynthesis, we have light dependent and light independent. So light dependent, we do have an enzyme. Which enzyme? NADP reductase, the enzyme that involved in the photolysis of water. We have these two. Are you clear? But the thing here is the entire process of the light-dependent reaction are more to the flow of the electrons and also the photoexcitation of the electron. Okay, so the impact of the temperature wouldn't be so great if you compare to the Calvin cycle. Because Calvin cycle, every single step involves enzyme. Are you clear? Calvin cycle, every single step involves enzyme. So if the question asks which pathway, the answer is light independent reaction or Calvin cycle. Okay, for temperature, always remember Calvin cycle. Okay. So as explanations of the effects here. So at low temperature, the rate of photosynthesis is low because of the Q10 effect. So what's the Q10 effect? Still remember the Q10 effects? So for any reactions that totally depend on the enzyme, that for example, Calvin cycle, for example, respiration, glycolysis, Krebs cycle, link reactions that totally depend on the enzyme, then you can see that the Q10 is almost equal to Q. Q10 basically means that for every increase of 10 degrees Celsius, the temperature, the rate of reactions will be double. For example, if 10 degrees Celsius, the rate of reaction equal to 5, then if I increase from 10 degrees Celsius to 20 degrees Celsius, you expect that if, let's say, that reaction totally depends on the enzyme, the rate of reaction will double up to 10. I increase further from 20 to 30 degrees Celsius. I expect that the rate of reaction will increase double. In this case, you will from 10 to 20, right? 5 to 10, 10 to 20. So if you look at the graph, now, you can see that the increase here is something like exponential. Slow and increase. Are you clear? It's make it a curve. Are you clear? Because of the Q10 effects for temperature. Okay, so the rate of the photosynthesis actually reached the maximum peak again at the optimum temperature about 35 degrees Celsius for this example. But plant usually 25 to 30 degrees Celsius. Okay, the rate of photosynthesis decreases when the temperature exceeds the optimum temperature. So this sentence actually also shows us that when you calculate for Q10 or you concept of Q10, one thing very, very important is the selections of the temperature. We always select 10 degrees Celsius, right? For example, 10 to 20. You compare 10 and 20. You compare 20 and 30. But you don't compare any temperature above the optimum temperature. Because any temperature above the optimum temperature is not valid for Q10 anymore because it's not increased. Right? You can see that the graph shows us the decrease. Correct? The decrease. So for Q10 effects only valid when the temperature that we choose to study lower than the optimum temperature. If you're more than above or above the optimum temperature, we don't calculate Q10, useless. Are you clear? It's nothing to do with Q10s anymore. Okay, and we do know that the shear active sites, again, Rubisco, for example, is no longer complementary to the shape of the substrate. In this case, carbon dioxide and RuBP, for example. I choose one step here. Okay, because of the denaturations. Am I too fast so far, guys? Okay or not? Clear? Here. Now, since we know that three factors, light intensity, CO2 concentration, and also the temperature will affect the rate of photosynthesis. For us, we see that thing, but for farmer or plantations company, this is very, very important because yield. How much products can be formed? If I can increase the rate of photosynthesis, means that the company will gain more profit because more yield. So one of the ways is to control the plant or crop plants. Where we grow them, how we grow them, and what types of the nutrients we need to give them. So this kind of environment is called protected environment. It's not like open air, but we can keep the plant inside the glass house. Are you clear? So we can actually supply artificial light, but make sure that you don't think that plant green color, they like green, but in fact they reflect green color. Are you clear? Okay, don't show, oh, my plant actually green color because they like green, so you shine a green color for I like to the plant. Basically, you are wasting your money. Okay, so we can actually supply the artificial lights. We can actually have the artificial heater, and also we can supply additional carbon dioxide. So that with these three factors involved, the company will earn more profits in this case because the plant will give more yield. Okay. But the thing here is, if you increase the light intensity, you could provide the heating and also the carbon dioxide. What happened here is you're going to incur the cost. So you cannot simply, okay, light intensity very, very high. Are you clear? We have to check. We have to get the balance. Okay. So at the same time, we can install a sensor so to monitor the light intensity, humidity, temperature, and even the concentration of CO2 around the plants. But we know that additional cost will be there. Okay, so you have to need to know the farmer need to find out the optimum growth condition. Okay, for example, guys, you look at the graph, rate of photosynthesis versus the light intensity, and then you can see it going up and then plateau. Can I see that? So it means that this is the max light intensity, because further increase of light intensity would help, but you're going to waste money. The farmer or the company need to know this limiting factor concept. If they don't know the limiting factor, simply increase the temperature, simply increase the CO2 concentration. At the end, you make a loss. Okay. So generally, in practice, we can actually use a paraffin lamp. Paraffin lamps, you can burn this paraffin lamp. So the burning of this paraffin will give us the additional light intensity, and the combustion give us CO2 and also heating process. So 3 in 1. But what's the disadvantage here? Can you control the temperature? You can't control temperature. Can you control the brightness? Cannot. It totally depends on this natural process when you burn the paraffin. But 3 in 1. Okay. So other advantages of the greenhouse or this glass house Basically, the plant can be grown hydroponically. We don't need to use the soil and land in this case, and we can actually control nutrient contents. We can maximize the yield by using the computers actually to adjust what kind of nutrients, what kinds of the cytokine, what kind of chemical we can add, fertilizer, so that the plant will grow optimally. Okay, and this kind of grass house also can protect the plant from the insect pests, fungal, bacterial infection, okay, or viral infection. Okay, so increase the yield.